The pathological factors affect the functions of organisms at every level: system level, organ level, and cell level. This lecture will present the effects of different factors on cell function and structure. Pathological factor may modify the function of the membrane in the cell, thus modifying the size and the shape of the cell. The membrane functions, such as the permeability, including the diffusion, osmosis, active transport, ion pumps, and also endocytosis and exocytosis. Also, to modify the interaction through the membrane with the environment of the cell. Different factors may cause cell injury. They are listed here: chemicals, physical agents, ischemia or hypoxia, immune injury, genetic defects, imbalances, and aging. They may be divided in three main groups. First group, deficiencies: energy deficiency, both oxygen or nutrient, primary, secondary, genetic defects, and viral infection. Here, the deficiency of nutrient is summarized. The primary nutrient deficiency is due to a lack of a nutrient into the diet. The secondary nutrient deficiency, the nutrient is present in the diet but it cannot be digested or absorbed. Genetic defects. They are related either to impaired metabolite regulation or impaired metabolic synthesis. So, deficiency state is generated and causes cell injury. Also, infection, especially infectious agent utilizing the needed nutrients, also may cause a deficiency state. Intoxications are another group of causes. They may represent as exogenous toxins, biological or non-biological, or endogenous toxin production, free radical formation in the organism, and impaired circulation. This is here a picture presenting the sources of endogenous toxins, for example, genetic defects. Most commonly, they result in defective enzyme functions, producing either an accumulation at high level of a normal metabolite exerting a toxic effect, or a direct toxin production, or an activation of alternate pathway causing the toxic injury. The impaired circulation may also contribute to the accumulations of high level of normal metabolites causing a toxic injury. The next group of causes, this is the cell trauma, including physical injury, thermal injury, ionizing radiation, mechanical pressure, microorganisms, and immune defects. The physical injury may cause directly a membrane rupture and loss of contents or organization of the cell. Thermal injury, both by hyperthermia or hyperthermia. In hypothermia, ice crystals are formed in the cytoplasmic water, frostbite. In hyperthermia, burning, for example by direct contact with a fire or exposure to excessive solar radiation, and also the thermic effect of current also may cause a trauma. Ionizing radiation causes trauma by burning at high doses or by disruption cells nucleic acids. Microorganisms, infection, bacterial toxins, and enzymes may cause breakup of connective tissues. Viruses may cause rupture of the cell membrane because of multiplication or sometimes direct attack, for example by protozoa. Malaria is causing cell rupture. And finally, immune defects. Antigen-antibody interaction at cell surfaces lead to a physical disruption of the cell membrane. For example, viral proteins incorporated in the cell membrane. formation of new channels or pores in the cell membrane, and destruction of the barrier function and the selective permeability of the membrane. For example, this is the effect of hepatitis B virus. This slide shows the major mechanisms by which the injurious stimuli affect the cell function. First, by causing a hypoenergetic state, decreasing the ATP production. Second, membrane damage, this affecting mitochondrial function, lysosomal function, and especially plasma membrane function. Also by increasing intracellular calcium level and by producing reactive oxygen species, active radicals. For example, the decreased ATP production may be caused by ischemia, affecting the oxidative phosphorylation in mitochondria. As a consequence, energy deficiency will affect the function of energy-consuming mechanisms, especially pumps into the membrane. For example, the sodium pump. The deficiency of sodium pump will allow sodium to accumulate into the cell together with calcium, and influx of water also will be produced due to osmotic gradient. As a consequence, cellular swelling, endoplasmic reticulum swelling, changes in the membrane, blebs formation, loss of microvilli, and so on. The deficiency of ATP will cause the switch on a glycolysis, thus producing energy by an alternative pathway. However, the anaerobic glycolysis is producing lactic acid, thus decreasing the pH of the cell. So, the consequence of this acidification this is shown here, damage to the nuclear chromatin and also decreased glycogen production and especially activation of some lysosomal enzymes. Other effects of the ATP deficiency is the detachment of ribosomes from the endoplasmic reticulum, thus affecting decreasing the protein synthesis. Let's now review briefly all these mechanisms. The membrane damage. It is caused, for example, by the decreased oxygen availability, the mitochondrial dysfunction, and especially the decreased phospholipid reacylation synthesis. This will interact with the membrane composition, especially loss of phospholipids and damage to the membrane. Also, the increase in cytosolic calcium will also produce activation of some enzymes, especially phospholipase, thus to increase the phospholipid degradation, lipid breakdown products, and damage to the membrane. And also, the elevated level of cytosolic calcium is stimulating the proteases, intracellular proteases, thus damaging the cytoskeletal damage and as a consequence also the membrane. Let's now review in more details the role of the calcium. The calcium is stored into the cells, into the mitochondrial and endoplasmic reticulum spaces. When released, it will cause increased cytosolic calcium level and secondarily the permeability of the calcium through the plasma membrane is increased. So, three sources of calcium will contribute for the elevation of the cytosolic calcium level. The consequences: stimulation of the adenosine triphosphatase and exhaustion of the ATP stored, activation of phospholipase, decreased phospholipids and damage to the membrane, and protease activation with disruption of membrane and cytoskeletal protein. And finally, stimulated endonuclease will affect the nucleus chromatin, damage to the cell nucleus. Let's now review the role of free radicals in the damage to the cells. Free radicals are produced as a consequence of some factors such as ionizing radiation, chemical oxidant, cancerogens, inflammation, reperfusion, and some chemical poisons, especially carbon tetrachloride. Here, the most important radicals are presented: reactive oxygen species such as superoxide, hydroxyl radical, hydroxyl radical, and hydrogen peroxide, and also carbon trichloride radical. Which are their effects? First, the most important effect, this is the lipid peroxidation of membranes, cell membrane and mitochondrial membranes. And second, protein inactivation, especially enzymes. And third important action, damage to the DNA. Free radicals are counteracted by some cellular antioxidant mechanisms, especially enzymes degrading, such as glutathione peroxidase, catalase, dismutase, and vitamin E and C. Here, this slide summarizes the generation under the effect of the already mentioned factors of radicals into the cell and their effect on the cells. Membrane lipid peroxidation, all membranes, plasma membranes, Golgi apparatus, lysosomes, mitochondria, and here the inactivation into the cytosol as a result of the action of superoxide dismutase, vitamin C, glutathione peroxidase, and so on. Neutralization, preventing the cell injury. This slide illustrates an important aspect of the formation of radicals, the self-propagation of free radicals, autocatalysis. After being formed, they interact with other molecules to produce a damage and to produce more and more radicals. Thus, this is a self-catalyzed chain reaction. How is it stopped? Free radicals interact to produce stable forms. Let me know that specific enzymes promote such reaction, antioxidant system, or another type of factors, scavenging radicals, forms also stable forms. This is the effect, for example, of vitamin E. The extent of cell injury depends on the nature of causative agent, also on the intensity of the causative agent or the duration of the causative agent, also by the number of exposures. As a consequence, either a recovery after an initial damage may be produced, or, if progressing, may cause the death of the cell by progressing of the both cytosolic and nuclear changes, and finally causing necrosis with rupture of the membrane and producing leakage of the content into the body fluids. The changes produced may be fully reversible. For example, the hydropic change or hydropic degeneration. It is presented here after initial damage and decreased energy production. As a consequence, sodium pump activity will be reduced and sodium will be retained into the cell, thus to increase osmotic pressure in the cell and to cause an osmotic influx of water, causing first vacuolation, later a phenomenon known as cloudy swelling of the cells, and the progression will cause finally a hydropic degeneration and even rupture of the membrane. The other change, also reversible, this is the fatty change, accumulation of fat droplets. Here, fat droplets are presented in liver and heart. Although reversible, the fatty change indicates severe injury. It is demonstrated by accumulation of fat droplets and swelling of the organ. Susceptible organs are the liver, heart, and kidneys, mainly caused by anoxia or intoxication, for example by alcohol. As an example, the accumulation of fats into the liver. This is here the turnover of lipids in our body, the adipocyte, plasma free fatty acid level, here the liver. The liver cells are metabolizing the free fatty acids or synthesizing triglycerides and assembling lipoproteins. Lipoprotein, this is the only form under which lipids are exported from the liver, thus generating a plasma level of lipoproteins. If blocked, either the metabolism or the production of lipoproteins, the fats will accumulate into the liver to produce this aspect, fatty liver or hepatosteatosis. Damaged cells may contain residual bodies. Usually the exogenous material entering the cell is engulfed and converted to a phagosome, fusing later with lysosomes, thus forming digestive vacuole. The content, after being digested, the remainder are expelled from exocytosis. However, in some cases, when especially organelles are damaged, lysosomes fusing will form finally autophagic vacuoles. However, some remainders of the membranes cannot be digested and they will stay into the cytosol under the form of lipofuscin granules. Thus, lipofuscin granules may be found as a mark of aging. This is a preparation of cardiac muscle tissue in an elderly person. Adaptive responses may include changes in size and cell numbers. For example, hypertrophy, cells or organ demonstrates enlargement that occurs in response to increased demands. For example, myocardial hypertrophy. Hyperplasia, this is another response. New cells are formed by mitosis to respond to increased demand. This is the response, for example, in epithelia. Atrophy or shrinking, this is caused by factors that reduce demand on a cell or decrease input stimuli. For example, the disuse atrophy due to decreased workload. Another adaptive response, this is the production and release of stress proteins. They are released under the impact of heat stress or heat shock. They are also known as heat shock proteins. Also, anoxia and toxins are able to produce stress proteins and formation of reactive oxygen species, and some attacks, for example, by the complement system forming a membrane attack complex. The cell responds by the production of stress proteins, which are able to bind to damaged proteins, thus to help enzymes to destroy them before clump formation. Also, to stabilize damaged proteins to enable them to become functional again, and finally to repair ion channels, to prevent apoptosis, to reduce oxidative bursts, and to suppress the production of pro-inflammatory cytokines, among other actions. This slide illustrates the effect of some factors causing intercellular depositions. This is the already shown example with the hepatosteatosis, the fatty liver, due to excessive production of triglycerides and deposition in the absence of opportunity to be exported from the hepatocyte, fatty liver. Another type of disorder, this is a protein mutation due to defective gene causing the production of defective protein, defective folding or defective transportation, thus causing its accumulation into the cell. Or another defective factor, a defective demonstration, lack of enzyme due to some genetic defect again, thus enzyme acting on a complex substrate to produce soluble product will not be effective. The substrate will accumulate into the cell and will damage the cell. And finally, ingestion of some materials that may be indigestible. Accumulation of these material will cause damage to the cell. This is observed in diseases such as anthracosis or asbestosis, accumulation of exogenous material into the cell and damaging of the cell. Let's now consider the forms of cell death. Apoptosis represents a programmed cell death. It is involved in normal cell deletion and renewal. It represents a controlled cell destruction. Apoptotic cell death eliminates cells that are first no more required, such as aged cells or embryonic cells during the embryonic development, also cells that have been produced in excess or cells that have developed some improperly type, or finally some cells with genetic damage, genetic defects. In normal cell turnover, the apoptosis provides the space needed for cell replacement. The apoptotic cell death may be defined as a controlled autodigestion of cell components. Cells initiate their own death through the activation of endogenous enzymes, which are the triggering factor for this process. All surface may be involved, for example, signaling by some presence of some factors or the lack of withdrawal of some factors such as growth factors and hormones. Receptor ligand mechanism, for example, between the Fas ligand and the TNF binding to a receptor. This is a so-called death receptor into a death domain. It triggers the so-called extrinsic death receptor initiated pathway. Other factors are stimulating the intrinsic pathway. For example, injuries such as radiations, toxins, free radicals, damaging the DNA and thus stimulating the activation of the factor P53. It is involved into the cell repair by stopping the mitosis, thus allowing the repair system to repair the DNA, or, if it is impossible, to stimulate the apoptosis by activation the intrinsic pathway. This effect is regulated by the BCL2 family genes. Let me now illustrate the effect of these two pathways in more details. They both will end by activation of the initiator caspases and later executioner caspases. Now, the extrinsic pathway. This is an example of the Fas ligand binding to the death receptor into the death domain and activated some other membrane protein to activate the procaspase 8. This is an inactive enzyme in inactive form. After being activated, autocatalytic caspase activation occurs to activate the caspase itself and also other caspases, executioner caspases, triggering the death of the cell. The other mechanism, the intrinsic, is started by the activation of the mitochondrion. As a consequence, content of the mitochondrion is released, especially the cytochrome C. Cytochrome C, in combination with other factors in the cytosol, will also activate the procaspases. However, this is another caspase, procaspase 9, when activated autocatalytically to produce activation of more caspases and finally to activate the executioner caspases and to produce the apoptosis. Now, the executioner caspases will stimulate a lot of enzyme, causing first destruction, fragmentation of the nucleus, and second, breakdown of the cytoskeleton, and also mobilizing the membrane in this process. The caspases are a group of cysteine-dependent aspartate-directed proteases, or In summary, cysteine aspartic proteases, the term caspase, is an abbreviation of the cysteine aspartic proteases name. In summary, caspases cause cell shrinkage as a result by disruption and clumping of the nuclear DNA and fragmentation of the nuclei, condensation of cytoplasmic organelles and disruption of the cytoskeleton, and finally distinctive wrinkling of the cell membrane and formation of apoptotic bodies. In fact, as the cell shrinks, the nucleus breaks into spheres and the cell divides into membrane-covered fragments, the apoptotic bodies. The membrane changes are signally surrounding phagocytic cells. They will engulf the apoptotic bodies and complete the degradation process. Thus, no inflammatory response is observed. To repeat, the physiologic apoptosis is involved and responsible for normal physiologic processes, cell destruction during embryonic development, hormone-dependent involution of tissues, death of immune cells, and cell death in proliferating cell populations. While pathologic apoptosis is involved, for example, as we will see in the next lecture, in cancer growth. Cell death by apoptosis is associated with certain viral infections, for example, hepatitis B and C, and also cell death caused by injury, mild injury, thermal radiation, and finally, apoptosis is involved in some neurodegenerative disorders, such as Alzheimer's and Parkinson's disease, amyotrophic lateral sclerosis, and others. This slide, a photograph illustrating a cell under apoptosis. The cell body is seen, the apoptotic bodies, including apoptotic bodies generated within the cell. Mildly damaged cells by a mild injury, despite the changes observed, may be subjected to recovery process and to recover to normal cells. However, if continuing the injury or increase in intensity, it will cause irreversible change into the plasma membranes, organelles, and nucleus till the fragmentation of the cell membrane and, as a consequence, death of the cell known as necrosis. This slide illustrates a case of so-called coagulation necrosis. The tissue architecture is preserved while the cell structures are lost. This is due to anoxic or toxic injury, usually. However, in the tissue, if the tissue architecture is destroyed under the effect of bacterial infection, bacteria stimulate phagocytes to release enzymes that will liquefy the surrounding tissue. So, this is known as liquefaction necrosis. Another case, unusual case of coagulation necrosis, is the so-called caseous necrosis, cheese-like necrosis observed in tuberculosis, or another unusual case of coagulation necrosis, the gangrenous necrosis, gangrene. It is due to noxious products of anaerobic bacterial infection in ischemic, oxygen-deprived necrotic tissue. This process is known as putrefaction, or gangrene, either dry or wet gangrene. Wet gangrene is accompanying the liquefaction because of the phagocytic action. This slide shows the progression of a typical coagulation necrosis into the myocardium. The injury causes necrosis of myocardial cells, but with the consequence neutrophils entering the necrotic area, later generation of inflammatory response with granulation tissue appearance, and also liquefaction and phagocytosis of the cell debris, and finally replacement of the parenchymal cells by connective tissue, fibroblasts, producing collagen and depositing fibers, scar formation. Thus we reviewed the two main types of cell death, the apoptosis and necrosis. When comparing these two types of cell death, which are the differences? For example, necrosis is caused, while the apoptosis is programmed. It is not caused, it is however either accelerated or decelerated by some factors. The apoptosis is starting by nuclear changes, following by membrane changes, while the necrosis is starting by membrane changes following by destruction of the nucleus. And finally, the necrosis causes an inflammatory response, while in apoptosis such a response is not observed. Now let me comment which are the consequences after a necrosis in our organism. Is there a repair after the loss of dead cells, the necrotized cells? First, in this case, there are no surviving parenchymal cells, total death. Let me note that the parenchymal cell of an organ, this is the main cell responsible for its specific function. For example, in the heart, this is the cardiomyocyte. In the liver, this is the hepatocyte. Total loss of parenchymal cell will cause replacement, repair by scar formation, replacement by connective tissue, production of collagen by fibroblasts, and formation of scar by fibrosis. This is the repair after total death of parenchymal cells. In a case there are sufficient numbers of surviving parenchymal cells, the response will depend on the cell type. Three main cell types are observed: labile cells, stable cells, and permanent cells. The labile cells are dividing even without stimulus. As an example, epithelia into the gastrointestinal tract. When damaged, they will repair by full regeneration, so the function will be fully restored. For example, if epithelia in gastrointestinal tract are destroyed, for example by alcohol, they will regenerate within hours or within a day. Stable cells, these are cells that do not divide spontaneously, but they are able to regenerate after damage and cell loss. However, under conditions of intact tissue, connective tissue framework. If the matrix is preserved, the stable cell will repair by regeneration. This is observed, for example, in the liver. Hepatocytes are an example of stable cell after damage, loss of parenchymal cells in hepatitis. Regeneration is fully possible in hepatitis if the intact connective tissue framework is preserved. This may be observed in acute hepatitis. However, in chronic hepatitis, especially aggressive hepatitis, the tissue framework may be destroyed, the connective tissue. So, in such a case, the repair will be done by scar formation. The chronic hepatitis will turn in liver cirrhosis. Scars will be deposited to form nodules into the liver tissue. And finally, the permanent cells, for example, cardiomyocytes, they are not able to regenerate. The repair after loss of parenchymal cells will be done by scar formation. Let's now comment the third form of cell death by autophagy. Autophagy is another form of self-destruction and self-death. Let me first compare with the heterophagy, the already commented response to a cell due to the engulfment of exogenous material by endocytosis or phagocytosis, thus forming a phagolysosome as a result of fusion with a lysosome, digesting the exogenous material and excreted by exocytosis of the remainders. In autophagy, there is an orderly degradation and recycling of cell constituents. For example, an organelle. Targeted cytoplasmic constituent organelles are isolated within vesicles, autophagic vacuoles, and later residual bodies will remain. Lipofuscin pigment granules are formed in that way. So, autophagy self-eating, it is observed under conditions of shortage of nutrients and oxygen. The exact value of this type of cell death in our pathology is not yet fully elucidated. I will now comment the vulnerability of different tissue types to injury. For example, ischemia. The central nervous system cells are highly sensitive because of the high metabolic rate dependent on glucose as their only energy source. They tolerate loss of blood supply for only a few minutes. For example, stroke is generated in that way. At second place, kidney or liver cells. Less sensitive, they can tolerate up to 60 minutes of interrupted blood flow without permanent damage. So, if cooled and decreasing energy demands, these organs may be transplanted after a transportation during up to 10 hours. The connective tissue, especially fibroblasts, they are more tolerant. They are able to form scar tissue at the injury site where oxygen level remains low until new blood vessels can form. Next factor, intoxication. For example, carbon tetrachloride. The effect of carbon tetrachloride is illustrated on this picture. When inhaled, carbon tetrachloride passes from the lung to the blood and finally to the liver cells. The greatest damage is observed in hepatocytes because of the breakdown of the carbon tetrachloride to a toxic free radical, trichloromethyl radical. The trichloromethyl radical is highly active. As already known, it will damage the membranes and as a consequence also polysome detachment and decrease the protein synthesis. Thus, the synthesis of lipoproteins will be affected. The fats cannot be exported from the liver, they will accumulate into the liver, thus fatty liver will be produced. In the other cells, the damage is not too extensive. However, the release of products of lipid peroxidation anyway, because of damaging of the plasma membrane and increase the permeability of the membrane to sodium especially, will cause influx of sodium and water and cell swelling, and the next influx of calcium, inactivation of mitochondria, cell enzyme and denaturation of proteins. So, cell damage, despite less expressed, is observed also in other tissue, but mainly in the hepatocyte. This is the sensitivity of the liver because this is the site of the degradation of the carbon tetrachloride and the production of the highly active trichloromethyl radical. Next factor to comment, this is the ionizing radiation. DNA is especially sensitive to high energy radiation damage. So, all actively dividing cells that are undergoing DNA replication are susceptible to the ionizing radiation effects. So, areas of rapid mitosis such as skin, red bone marrow and gastrointestinal mucous membranes present the greatest disruption of function after exposure to radiation. Thus, in the skin, hair loss will be observed. In the bone marrow, suppression of hemopoiesis. In the gastrointestinal tract, diarrhea, gastrointestinal hemorrhage, nausea. The next factor to comment, This is the viral infection. A specific interaction between the virus and the host cell is involved, so that each virus can infect only a specific type of cell. Susceptible cells have membrane surface receptors to which the viral particles can bind, the critical step that allows the virus to enter a specific cell. For example, the poliovirus. This virus infects neurons in the gray matter of the spinal cord, the anterior horn cells. However, several other types of neurons immediately adjacent are spared. They are not affected by the poliovirus. It remains to comment one more aspect of the cell injury caused by some agents such as chemicals, radiation, and viruses, producing DNA damage and cell mutation, producing mutated cell. This is the initiation of a pathogenic mechanism ending by formation of a malignant tumor. Because of the importance of this phenomenon, it deserves more attention and will be commented during the next lecture. Now, to end the lecture today, I will comment briefly how is it possible to determine and to monitor the cell injury in the whole organism. For example, it is possible to evaluate the functional loss. For example, the increased levels of plasma bilirubin in our body fluids are due to damage to the liver and destruction of hepatocytes, thus releasing the contents of the cell into the body fluids, hyperbilirubinemia. It demonstrates damage to the liver cells. Decreased proton excretion may be documented by monitoring the kidney function. Damage to the cells into the kidney tubules is demonstrated by changes into the pH of the urine. Damage to lung cells may be demonstrated by alteration in the partial pressure of the CO2 and oxygen into the blood. Another parameter able to show damage of the cells, this is the release of some cell constituents. For example, the plasma level of potassium, if increasing, this shows a cell rupture, widespread tissue damage, for example hemolysis, releasing cell content with high concentration of potassium. So, the increased level of potassium demonstrates a tissue damage. Increased plasma level of enzymes, for example creatine phosphokinase. This enzyme is abundant in cardiac, muscle and brain. So, this elevation of its level into the plasma is indicative for stroke or heart attack. Another enzyme, gamma-glutamyltransferase, when increased, will demonstrate liver damage. Increased level of troponin T and I is demonstrating damage to the cardiomyocytes and release of their content into the body fluids, or this is the evidence for myocardial infarction, ischemic necrosis of cardiomyocytes. Another approach to show and monitor cell injury is the registration of electric activity. For example, in electrocardiography, electroencephalography, on electromyography. And finally, damage to the cells may be proved histologically by analyzing biopsic material.